Hey, my name is Mary Rolfe, and today I'll be talking about the determination of sex, genes versus hormones. So let's go ahead and get started. Okay, so first I'm just going to go ahead and start off with some background information about the determination of gender. Okay, so just a few things that are important to know as far as background information. The human genome programs sexual development. Humans have 23 pairs of chromosomes, or 46 total. That can differ based on different genetic disorders, but typically this is what is ideal. The genetic sex of an individual depends on the sex chromosomes. Females have 2 X chromosomes, and males are XY. So in the absence of a Y chromosome, a female will develop from an embryo. Male and female sex organs consist of gonads, internal genitalia, and external genitalia. As far as internal genitalia, this is accessory glands and ducts that connect the gonads with the outside environment. And then the external genitalia are all visible external reproductive structures. So gonads can be classified as male or female gonads. Male gonads are called testes and female are called ovaries. Okay, so genes versus hormones. So the determination of sex depends on the presence or absence of the sex-determining region of the Y chromosome, which is also called the SRY gene. If it's present and functional, the bipotential gonads will develop into testes. But if the SRY gene is absent, the gonads will develop into ovaries. This is under the direction of multiple female-specific genes. And if you look at this image on the right, you can see it says this gene is Y-linked because it is only found on the Y chromosome. So, male embryonic development. The SRY gene produces a protein called testis determining factor, abbreviated as TDF. This TDF protein binds to DNA, and then it activates additional genes. The protein products of these and other genes direct development of the gonadal medulla into a testes. Testicular development does not require male sex hormones, such as testosterone. An embryo cannot secrete testosterone until after the gonads differentiate into testes. But 3 hormones are secreted from the testes once they differentiate. And then Sertoli cells secrete the glycoprotein anti-Mullerian hormone, abbreviated as AMH. And then the testicular interstitial cells, also known as Leydig cells, secrete the androgens testosterone and dihydrotestosterone, abbreviated as DHT. And if you look at the image on the right, you can see the different pathways that differentiation can take. The left side is the male embryonic development pathway, and then next we'll go over the female pathway. All right, so female embryonic development. The cortex of the bipotential gonad develops into ovarian tissue, and then, like I said before, multiple genes are required for the development of the functional ovaries. So in the absence of testicular AMH, which is the anti-Mullerian hormone, the Mullerian ducts will develop into the upper portion of the vagina, the uterus, and the fallopian tubes. And then without testosterone, the Wolffian ducts degenerate. And then without the dihydrotestosterone, DHT, the external genitalia take on female characteristics. All right, so these are just some stages of embryonic development in terms of weeks. So for a male embryo at 6 weeks, the gonadal cortex regresses, and in the female, the gonadal cortex will form an ovary at 6 weeks. The gonadal medulla in a male embryo will form a testes, and then in the female, the gonadal medulla will regress. Male 6-week embryo, a Wolffian duct forms the epididymis, vas deferens, and the seminal vesicle. And then, on the contrast, the female 6-week embryo the Wolffian duct will regress, and the Mullerian duct will form the fallopian tube, uterus, cervix, and upper portion of the vagina. But in the male, that duct will regress. And then whenever you look over to the 10 weeks stage of development, in the male embryo, the SRY protein will direct the medulla of bipotential gonad to become testes. And the AMH, or anti-Mullerian hormone, causes the Mullerian duct to disappear in the male embryo. And then in the female embryo, the gonadal cortex will become the ovary, and the Wolffian duct disappears in the absence of testosterone. So I'll just review some of this information. If the embryo is male at 6 weeks, the gonadal cortex will regress, the gonadal medulla will form a testes, Wolffian duct forms epididymis, vas deferens, and seminal vesicle with testosterone present, and the Mullerian duct regresses because AMH is present, the anti-Mullerian hormone. But in a female at 6 weeks, the gonadal cortex becomes an ovary instead of regressing. The gonadal medulla will regress. Wolffian duct regresses because the testosterone is absent. And then the Mullerian duct becomes the fallopian tube, uterus, cervix, and upper portion of the vagina because AMH is absent. And then over onto the 10-week mark, again, 10-week male embryo, the SRY protein directs the medulla of the bipotential gonad to develop into testes. And the AMH hormone from testes causes the Mullerian ducts to disappear. But in the female at 10 weeks, the gonadal cortex becomes the ovary in the absence of the SRY protein. And this is also, like I said earlier, under the influence of several female-specific genes. And then the absence of testosterone is what causes the Wolffian duct to disappear. So we can go ahead and move on to the review sheet that I made. Let's see. So, let's see. This is just matching terms to the function. So the key is below, but chromosome pairs in a human genome, that's going to be answer choice B. There's 23 pairs with 46 chromosomes total. Number 2 is the cause. Which of these causes development of testes from the bipotential gonad? The answer to this is the SRY gene, which is answer choice C. Number 3, what secretes the anti-Mullerian hormone? That's the Sertoli cells, so D. And then number 4 secretes testosterone and dihydrotestosterone. That is the Leydig cells, H. Number 5, the absence of this causes external female genitalia to develop. That is the dihydrotestosterone, or DHT, so that's E. And then number 6 is the absence of this causes Wolffian duct to regress. That was what we talked about in the last slide a lot. That is testosterone, so that's F. 7 is absence of this causes Mullerian duct to develop into the upper portion of vagina, uterus, and fallopian tubes. The answer to that one is the anti-Mullerian hormone, so G. And then allows for the development of prostate gland and external male genitalia. That's going to be the dihydrotestosterone again, which is answer choice E. And then the last one, the absence of this causes the gonads to develop into ovaries, the bipotential gonads to develop into ovaries. That's the SRY gene again, so that's answer choice C. All right, and that's everything.